Hi, I'm Sean Mooney, professor at the University of Washington and Chief Research Information Officer of UW-Medicine. Over the past 2 years, we have been creating an initiative in medical data science, and as part of that, we have created this seminar. Speakers will include world-leading scientists who will tell us about how data can impact human health and their healthcare. Please use the link below to register for our mailing list to hear about future events in this series. We hope you enjoy them.
Yeah, so we are really excited to have Professor Suen Lee, who is a Paul G. Allen Professor in the School of Computer Science and Engineering here at UW. She is also the Director of Computational Biology and AI in Healthcare Programs. Before joining UW in 2010, Professor Lee was a visiting assistant professor in Carnegie Mellon in the Computer Science Department. And she has received numerous awards like the NSF Career Award, NSF Innovation Award, and she's also been named as American Cancer Society Research Scholar. Her research particularly aims at integrating AI and medicine to make novel discoveries from patient data and molecular data. We are really excited to have you, Professor Lee, with us today and hear more about your work. Her title of the talk is Explainable AI, Where We Are and How to Move Forward for Cancer Pharmacogenesis. Over to you, Professor Lee.
Okay, so let me share my screen. Okay, is my slides shared?
Yes.
All right, thank you so much. Thank you so much for your introduction and then for the invitation. So what is the talk today about? So as complex black box modern machine learning models, such as neural networks, are increasingly adopted as integral parts of the data analysis in biology and medicine, model interpretation has become a new challenge. So in this talk, I will go over a number of research projects done in my lab on the topics of explainable AI applied to biomedical problems. So the main message will be that explainable AI can help us make new biological discoveries from data, make informed clinical decisions, and can even open new research directions in biomedicine. And however, it will need to evolve and then improve to be able to really solve real-world problems in cancer, in computational biology and medicine in general. So today's talk will consist of two parts. In the first part, I will give you a high-level overview of many examples of what explainable AI can do to address new scientific questions and open new research directions in biology and medicine, which we can't do otherwise. And we will see that explainable AI is no longer a luxury. So in the second part, and we are going to have a deep dive into our paper, which is under review. And I'd like this talk to be a way to convey to really and then interact with you on this question of what we really think and then discuss together how this field of machine learning interpretability or explainable AI needs to grow and evolve to be able to solve these problems in biology and medicine. So one of the goals is that after this seminar, I'd like you to all go back to your research problems and then apply this line of techniques I'll be presenting to interpret your own machine learning models and can do and then do things that otherwise is possible. Okay, so first of all, what do I mean by explainable AI? To show you one form of explainable AI, I am going to use a very simple motivating example. Say that we have a set of patient features and a well-trained machine learning model. We can predict various kinds of clinical outcomes or clinical phenotypes. However, in traditional machine learning paradigms, we can't really answer a very natural, basic question of why a certain prediction was made. And then the reason for this challenge is that with modern large data sets, the best accuracy is often achieved by complex models, which even experts struggle to interpret. And then the question is how to overcome this accuracy versus interpretability trade-off. And to resolve this challenge, we have developed a series of machine learning techniques to estimate each feature's in importance for a particular prediction made on a particular patient. So the first method we named the SHAP, SHAP, is a general machine learning framework that can be applied to any model. It doesn't matter whether you use tree ensembles or deep neural networks. And then it has 3 desirable theoretical properties, which other methods in this space do not have, and they made it one of the most popular feature attribution methods. So SHAP enabled us to understand how to determine individualized risk factors of kidney, chronic kidney disease and all-cause mortality. And then we also showed how to diagnose errors in a prediction model when deploying the model to predict, for example, say, surgery duration by explaining the prediction error. So in this problem, you don't explain your prediction, you are explaining the prediction error when you deploy the model. So similar black box challenges occur when we use a black box complex model, machine learning model, to understand the relationships between molecular profiles, such as gene expression data in brain tissues, and then neuropathological phenotypes, such as A-beta, tau, or Braak stage, and various measures of Alzheimer's disease severity. And then this model may predict the phenotype accurately, but does not give us mechanistic explanations of how gene expression levels influence or are associated with the phenotypes. Machine learning model is basically a black box. So to show you an example from our recent publication, which is co-senior authored with our faculty, Sarah Mostafavi, when we apply this multi-tasking deep neural network to more accurately describe the complex relationship between gene expression levels and then 6 neuropathological phenotypes, SHAP can be used. It can compute the individual genes contribution to each neuropathological phenotype, even when we use this complex black box neural network model. And one of the advantages of these models is the ability to detect non-linear relationship. For example, how genes are associated with the phenotypes depending on the sex. So which in our paper led to a previously unknown molecular mechanisms underlying sex-dependent neuropathology. So this was enabled by the use of nonlinear modeling between gene expression data and phenotype. And then we can still find the relationship between each of these neuropathological phenotype and the individual features. So we have also shown that Explainable AI can also help make informed clinical decisions. So the SHAP method helped us develop the prescience system to predict the near-term risk of hypoxemia during surgery and then explain the factors that led to that risk. So, and then we showed that in our paper, the explainable prediction of hypoxemia could improve clinicians' ability to predict this event. Obviously, it's an unwanted physiological condition, and then being able to predict these events more accurately means a lot. So just, this explainable, predictions can also provide a new way to use this machine learning for biomedicine. So all this example project I just briefly showed you is in these examples, I showed that, when you can explain the prediction based on input features, you can already identify some biological discoveries or improve health, the clinical decisions. But then the question is what to do, further. How do we evolve? So for example, recently we developed a model auditing framework for clinical AI by using this explainable AI techniques. So, and then in our paper recently published in Nature Machine Intelligence, we revealed that many published clinical AI systems learned a shortcut rather than genuine pathology from X-ray images. So in this paper, we basically stopped. The field moved to the wrong direction. And then there were already 6 published papers and hundreds of related models out there that learned the shortcut. So what is this about? So we began with the hypothesis, which became our main conclusion of the paper, that the published AI systems that detect COVID-19 relied on shortcuts to a larger extent you wanted than genuine pathology. So then the source of the problem turned out to be in the way the training data were generated. So for the published models, a lot of published models that we referenced a bunch in our papers, in our paper, they all constructed their training data in essentially the same way, which is by combining COVID-19 negative images from one data source with positive images from another data source. So, and then when we created this generate data number 2 external to this training data in a different way, say, it has a positive and negative classes collected over different time periods in different sets of hospitals, but by the same research team from the same geographical region, we can then do cross data set experiment where you have here data set number 1 and 2. Each of the data set, you split them into training and test samples. And then you can not only test your model by using one of the data sets, using training and test split, but also you can test, you can use the test images from the other data set. So, but then, this, when we compared the performance by using this, ROC curve in 2 situations where you tested internally, the red colored versus externally, the green colored, this, what it shows is that the prediction performance deteriorates significantly. So this, illustrates a potentially dangerous situation in which the algorithm appeared highly accurate, but it fails when tested in hospitals external to the training data. So when you swapped the training and test data, so say you train your model using data set number 2, using the training images, training sample portion, and then compare and then test in using the test data set number 1, you see similar phenomena. So and then to understand why these models failed to generate, we took a look at the saliency map to explain the prediction. So I'm just showing you one of many examples. In our paper, you can see a lot of such examples. So for example, it is right that the model used a clear long basis as a way to conclude that this particular sample is not COVID-19. But it is not right that the model used this, laterality marks or, something else, or even, the device that the patient was wearing. We certainly don't want the model to pay attention on these parts. So, and then we have done a series of analysis starting from this. So this is explainable AI. You are attributing your prediction to input features, and then we also presented many other ways to detect that many of this the published models learn the wrong things, the shortcut. Okay, so now the next topic, I'm gonna show you a project on using the feature importance to help saving the feature gathering cost. So this was the collaboration with the emergency medicine department led by Dr. Nathan White, and then the first author of the paper was Gabe Arian, who just got the PhD from the MD PhD program. So we, in this paper, we demonstrated that we can use a SHAP to make a medical AI model cost aware. So, and we developed this co-AI framework, cost aware AI framework. And then the idea is that in many clinical applications, gathering cost is often costly, and then in many of these medical AI projects you are seeing, we pay more attention on prediction accuracy rather than feature gathering cost. So, but if you think about application like, predicting ATC, the acute traumatic coagulopathy, which is a very dangerous bleeding disorder that happens in a trauma patient, then first of all, it is very time sensitive. Not only it is important to predict accurately whether the patient is going to have ATC, it is also important to diagnose or predict fast. So because it often requires a massive blood transfusion, and an earlier transfusion leads to better outcomes. So in this case, the cost would be time. So our goal is to predict coagulopathy before even the patient arrives at the hospital with little feature gathering cost. And then here is the prediction performance plotted against the cost of the the feature gathering cost in the x-axis. So now in this framework, you are going to measure the predictive accuracy within varying range of model cost. So we use the Harborview Medical Center trauma registry data, and then we train the model. And then we plotted the prediction performance in the cross-validation, separate training and then test split situation, and then plotted the prediction performance against varying levels of a feature gathering cost. So here we worked with many experts, domain experts, EMS providers, to get a sense of how much it takes to measure and obtain each of these features, and then we compared, and then Co-AI, our framework, worked significantly better than many other alternatives. One of the alternatives is the PACT score. It's a previous published approach that takes 6 features, and then it takes 8 minutes in total to gather all these features. And then you can see that with the same prediction performance, you could get better predictive performance with much cheaper features, in this case in a more time efficient way. So Co-AI is a general framework, can be applied to other kinds of costs. So one example we presented in our paper was that we try to predict inpatient mortality in 200 US intensive care units data from MIMIC project. And this plot, the same kind of plot, area under the ROC curve in the y-axis, and then it's plotted against the x-axis, the model cost. In this case, the cost will be the number of features. And then we compared the Co-AI framework with a number of scores that are already used at ICU as a way to predict the mortality of the patients. Okay, so there are a couple of examples that, and then you can find in our paper, which is in press, but it's a meta archive version should be available. Okay, so now a natural question is what to do next. So do we need to improve these model explanation methods in a way to more accurately compute feature attributions, and then develop better methods for that? And then to get an idea of how to improve, it may be important to understand how all these existing methods work. So in our recent publication in JMLR, we showed that we can explain 26 published popular explanation methods within a very simple, unintuitive, very intuitive unifying principle. So it was led by my student, Ian Covert. So what is this about? So we propose a unifying principle we called explaining by removal, which means that explaining how a model works by removing the influence of groups of features or individual features, and then observing the impact it has. So this unified theory can cover at least 25 existing methods. And then with this perspective, rather than seeing this monolithic algorithm, so we can see that all these methods are built on the same foundation using interchangeable choices. So Yeah. So what is this? What does this framework say? So removal-based explanations are, described by a simple framework, and then it says that the recipe for making one of these methods involves making 3 choices. So first is that how to remove features from the model. So to turn off a feature's influence and then see the impact that has on the model. You want to just not give the model access to that feature. That's not how machine learning model works. So, but you can really not give this access because you need all values of all features. So you need some trick for simulating feature removal. So you may set the remove the feature to default value, neutral value, or you may say if I don't know this value, then I would just simply average over the distribution of the values. So the second choice is what model behavior do you want to explain? So as features are removed, you can see the impact it has on the model, but what quantity do you want to observe? Perhaps it can be the individual prediction or loss for an individual prediction, or just perhaps the data set population loss. So the third one, lastly, you need a summary technique, and then you can remove any set of features and then see the impact, but that's an exponential number of subsets. It's too much information to communicate to a user or model developer. So a summary technique provides a way of condensing that information into something more manageable, like single score for each feature or a subset of the most influential features. So we did all these 3 choices, and there are many options for each of them, and then you can design a ton of different explanation methods. So tomorrow you can develop a new explanation method. So we found that more than 25 different methods fit into this framework, and then this table, each row is a method, and then the entries show the choices they make for each dimension of this framework. And our framework can cover local and global methods and the feature attribution and the feature selection methods as well. So and it may come as a surprise, but this shows that many state-of-the-art methods are constructed using interchangeable choices and then simple unifying theory, which is a feature removal. Okay, now in the second part of this talk, I'm going to have a deep dive into our paper that's currently under peer review, Nature Biomedical Engineering. So, and then the first author is our MD PhD student who is defending today, by the way, Joseph Janizek. And then this is collaboration with Kamila Nexarova at Harvard and MGH. Okay, so so I just told you all these successful stories about, how explainable AI can advance our knowledge in biology and medicine, and then help make informed clinical decision fast and accurately. And then, now what I'm going to talk about now is the challenge. So, this is an example where you can see applying the current best explainable AI method is only the beginning of the story. And then you will immediately find some really, big challenge. So to give you a bit of background on the problem in general, the cutting edge in the treatment of cancers like blood cancer, we focus on acute myeloid leukemia in this project, has increasingly become combination therapy. And it makes sense because, by choosing drugs that, target complementary biological pathways, we have a better chance to achieve, better anti-cancer efficacy. And then that means to cancer patients, not only better chance of survival, but also with a better quality of life. So, but the issue is that choosing optimal combinations of drugs is a hard problem. There are already hundreds of individual FDA-approved anti-cancer drugs, which means that there can be tens of thousands of possible combinations if you only consider pairwise combinations. And then there could be even more if you consider non-FDA-approved experimental drugs in development, or consider combinations of more than 2 drugs. So finally, different patients, even patients who have the same type of cancer, may respond differently to the exact same drugs due to particular genomic characteristics. So if you know machine learning, you first thought that, that might be it sounds like a perfect opportunity for predictive machine learning based modeling. So you could collect a data set with a bunch of samples, including the characteristics of different patient AML tumors, such as gene expression levels, and the characteristics of a bunch of different combinations of drugs, such as structure of these drugs or their biological targets, their gene targets, and use these data to train a machine learning model that will generalize well, so that it can then be used to predict how synergistic different combinations of drugs that you never tested will be for different patients. And then so we attempt to do exactly this using a data set consisting of tens of thousands of samples made up of 133 two-drug combinations tested in nearly 300 patients. So, and what we found was that complex model, again, tended to outperform linear models. And then in particular, a class of models based on decision trees called XGBoost or gradient boosting machines, GBM, outperforms all the other methods classes. So each dot here is a comparison between two models trained on one cross-validation split of the data. Y-axis showing the performance of the GBM model, gradient boosting machine, and then the x-axis showing the performance of the other model we compared with, including elastic net, linear model, and the neural network, and the random forest tree. So, and then, we know already from the work I just mentioned on this COVID-19 detection with the neural networks, how important the model interpretation can be in the context of clinical applications of machine learning. So in some sense, interpretability may be even more important in the context of a pharmacological development, where the discovery of a particular mechanisms and interactions underlying successful therapies can motivate future research into new and then more effective drugs. So in trying to apply a feature attribution methods like SHAP to the high-dimensional gene expression data sets, we ran into an interesting challenge. So to measure how well methods such as SHAP rank features compared with the more traditional linear models for feature selection, we looked at three types of data structure. So a synthetic data set with the totally independent normal features, normally distributed features, and a synthetic data set with the groups of tightly correlated features, the middle one, and then the last one, the real gene expression data from AML patients. So, and then we defined the synthetic labels for these data set using nonlinear function of randomly selected features, and then measured how well different methods ranked features compared to their true data rankings. So in the first two cases, we see that nonlinear models such as XGBoost and neural networks combined with the SHAP outperform other approaches. And however, as a feature correlation increases, and when we use real gene expression data, we see all methods do not do well, including the nonlinear model with SHAP. So feature attribution methods such as SHAP are not robust when features are correlated to each other, because its assumption is a feature independence. Basically, correlated features will split the credit. So, these feature correlations make model explanations even more confusing. So when we trained this XGBoost model on the same data set with different random seeds, or you can view this as neural networks with a different random initialization, we found that these models that got around the same test error ranked the features very differently. So each row in this heat map is a model trained on the same data set. Each column represents a feature while the intensity of each cell encodes the importance of that feature. So those of you, with a background in machine learning or statistics, this is a phenomenon that's called rational effect. So, or, multiplicity of good models. So what it means is that, when there are many, it refers to a situation when there are many explanations exist for the same phenomenon. So how should we then choose which of these similarly performing models that provide different explanations? And put another way, which of these models will have explanations that best reflecting the underlying true biology? So to get an answer, we generated synthetic labels for this AML data so that we could see how well the attributions matched the ground truth. And then we saw something even more concerning. So y-axis here is the measure of how well the model, how well the model's feature ranking aligned with the true feature ranking. And the x-axis is the prediction error. So we see that, while there is a strong anti-correlation overall, which is what we wanted to see, between error and the feature ranking performance. Within the same model class, there is no significant correlation between test error and the feature attribution quality. There are basically 3 blobs, each corresponding to each model type. So this means that simply picking the one model with the lowest test error may not reveal the most biologically important mechanisms. So we came up with then a simple hypothesis. Instead of choosing a single model's explanations to consider, we could instead ensemble the attributions over many of these well-performing models with different attributions. So, and these ensembles of complex, these GBM models are then, what we applied to our final analysis. And this is something we can use when we use these explanation methods for genomic data, where often we see lots of correlated features. So once we have this more stable and ensemble attributions, we can now see how interpretability allows us to do sanity checks on our model's predictions. So one example is that we consider the feature attributions for drugs venetoclax, a small molecule that specifically targets the gene BCL2. And then when we looked at the set of genes that are important to the model's prediction of synergy in combinations that contains venetoclax, we find its actual target BCL2, as well as many closely related genes known to interact with BCL2. So furthermore, we can now then look at what genes were important across many different drug combinations in our data set. We find that 2 of the most globally important drugs are MEIS1 and then DLL3. So both of which are very well been linked to AML prognosis, and then both of which are related to cancer levels of differentiation, or how much that cancer retains a pluripotency stem-like state. So, and then the importance of this cancer stemness is further confirmed when we checked the biological pathways that are enriched with the genes from the top of our list, most predictive, most important genes. And they find that a pathway containing genes marking a cell state of differentiation or stemness within the hematopoietic cell lineage is one of the top pathways. So to verify that the extent of a cancer's stemness or differentiation really was generally linked to drug synergy across many combinations, we used purified, totally separate set of purified expression profiles from different blood cell populations to create expression profiles directly relating to a blood cell's state of differentiation. This led to differentially expressed gene sets between 6 pairs of stem-like and then differentiated, purified hematopoietic cell populations. So you can see there are, using this six comparisons, you can create 12 gene sets. We then calculated the average expression of the genes in each of these profiles for each sample, and then measured the correlation between synergy for each combination of drugs, and then the average expression of each profile. So here a profile means, say S1 means the genes that are, when you compare between stem-like hematopoietic stem cell versus more differentiated cell, the genes that are highly expressed in stem-like cancer. So that leads to a set of genes. So that's S1. Okay, the other way is D1. So and then here we then cluster this, these drugs based on the similarity of their correlations across the profile. So here again, S1 through S6 corresponds to gene sets that are more highly expressed in stem-like cells, or D1 through D6 would be the gene sets that are more highly expressed in differentiated cells. And then we, for each of these gene sets, we computed the average gene expression levels, and then calculated the correlation between these average expression and then the synergy between the average expression and then drug synergy for all drug combinations in our data set. And then you are seeing the list of drug combinations that resulted in statistically significant correlation. So some of these combinations are well supported by recent literature, which was really exciting. For example, ruxolitinib plus venetoclax, a combination known to be synergistic broadly in many patients with AML. It was a recent literature that suggests that showed that. And then let me see. Yeah, and so what would be the underlying theory on that? So for example, this more monocytic clones of AML are known to be more resistant to venetoclax. And then what that means is that what our results support is the theory that this ruxolitinib can overcome venetoclax resistance in these more monocytic cancers by either in two scenarios. So first is either sensitizing the cells to venetoclax or killing the subclones of the cancer that's not killed by venetoclax. So and then while this particular combination is already known to be a promising combination, our approach identified several other drugs that may be promising in combination with the venetoclax due to the same mechanism. So our approach also identified completely novel combinations that do not contain venetoclax that can also potentially attain synergy through the same underlying mechanism. So either altering the transcriptomic programs of differentiation or targeting different kinds of subclones at different stages of differentiation in complementary manner. All right, so in this talk, we went over many examples of research projects. In the first part, we went over many examples of what explainable AI can do to address new scientific questions and then even open new research directions in biology and medicine. And then in the second part of the talk, we had a deep dive into our ongoing paper, under review, which is on cancer pharmacogenomics. So I'd like to thank everyone in my group. And then as you have seen some small portion of the research project we have done, our recent research focuses on a broad spectrum of problems, including developing, interpretable machine learning techniques. And then in basic biology side, we are interested in identifying cause and treatment of challenging diseases such as cancer and Alzheimer's disease. And then clinical AI side, we are interested in building or auditing clinical AI models. And then our lab, AIMS lab, is a group of passionate researchers consisting of PhD students in the Allen School, and then a couple of MD PhD students who are pursuing a computer science PhD program. Okay, thank you so much. And then I'll be more than happy to take any questions you might have.
Thank you, Dr. Lee, for that engaging and interesting talk. We don't have a lot of questions in the chat, but I'll let people unmute their mic and ask questions if they have any now. Please, if you have questions, just unmute and you ask. Other than that, I don't see a lot of questions in chat.
So this is Dushyant Sahani, Chair of Radiology. One question, impressive work. My question is related to platform for to deploy, the models for, testing the models. What is your view on the platform? What platform do you use for that?
Yeah, so we haven't really, deployed our system yet, and then that's obvious. The one of the next thing we would really like to do. So by platform, well, I guess it can depend on the application. Like radiology, like yourself, or anesthesiology or emergency medicine. So, but I think in general, this kind of a platform really needs to have not only this machine learning system, but also a mechanism of auditing it to make sure that it learned the right things, and then also you can explain, why the model deteriorates over time if it happens.
Thank you, Dr. Sahni, for that question. So now Dr. Harefield actually is asking, are your models assisting in making patient treatment questions that involve thresholds and branching decision points?
Yeah, that's also another thing, as a future research that we need to move forward. Right, so not only the patient, we also need a way to interact with experts as well. The ideal explainable AI system would have something, some way to interact with the humans, especially in this case, it would be clinicians, the domain experts. And yeah, to answer your question, we haven't yet. We haven't built this, assisting, the model that assists the patient directly yet. That's exciting next step. What we established now is proof of concept.
If I could clarify the question, sorry, I was typing too fast and it's not grammatical, but it seems like when you're making decisions, particularly something as complex as chemotherapy treatment for a patient, there's going to be threshold effects where above a certain level, a patient is not going to respond, or there are optimal sweet spots where patients may respond to one chemical if it's been shown that they failed to respond previously. And physicians are making decisions about, well, I've reached this point. How can your system help me make the next best recommendation based upon these previous responses? And it seems like your systems are incredibly powerful, but without, I think they call it first order logic, yes, no, if then. How can a neural network navigate the very complex threshold and branching decisions required in patient care?
Yeah, I think I know what you're getting at. So that is why there is a lot of room for research. A lot of research needs to be done because machine learning is very powerful. We see a lot of all these great things machine learning or AI can do, but still it takes a specific set of input and then output, and it should be trained exactly in the same setting. So that's one area where we need to grow in terms of this core ML and then AI.
Thank you.
Hello, Dr. Lee. I have a question.
Sure.
Really impressive work, so thank you. My question is about how relates to the causal inference. So how can I, you incorporate your framework for understanding the causal mechanism of a biological disease? Yeah.
Yeah, that's an excellent question because the explanation is not causality. And and it's just close to say, when, you must, we have heard a lot about this, right? Association is not causality, right? I mean, it's what you see from your, statistical association, never causality, the same story, same thing. So, whenever, someone asks me, what is your next? So you have all these feature attributions, a perfect feature attribution. What is your next in terms of the explainable AI research? I think causality is one of the questions that is really one of the most important questions next. Because we, biomedical researchers, we always want to find causal. I mean, if you're a basic biologist, you want to find causal mechanisms, which gene regulates which other genes. And then if you are a clinician or, medical scientist, you want to know which factors intervene, which factors actually has a causal impact on the clinical outcome. Say it's like adverse clinical outcome you want to stop, right? So, and then, it's the next step. In terms of how to go from attributions, the explanations to causality, there needs to be some extra information because from observational data, you can't really identify causal relationship. So such as, known knowledge. It is already known that, gene A regulates gene B, or, A is a genetic, there's a DNA. So it has to be causal, not defect. Something like that. So there needs to be, this ML algorithms need to be developed to properly incorporate prior knowledge or, domain expert knowledge in terms of to actually, get to the answer of what really caused.
Thank you. That's really helpful.
I actually have a question about the talk. So you mentioned how these AI systems, where the explainability from them could be used to sort of like guide like knowledge about the system. So for example, in the drugs, combinations of drugs that the AI found could be then used to guide knowledge about how those drugs work together. And so far, it's been a story of knowledge driving AI in terms of feature engineering and feature selection. Do you think that that's a potential direction that medicine is going or could go in the future, where the harnessing big data and AI could be used to actually guide knowledge and as well as like advance?
Yeah. It has been used that way, right? Yeah, machine learning. The way machine learning is applied to this biological data. So in some sense, it's like a tool to extract knowledge from big data, and then the result we see is through this lens of a machine learning model. So that is why advancing machine learning models is so important, because that can shape our knowledge. Okay, Dushyant.
Thank you, Suan. One question is, where do you see potential successes on the medicine side, like whether it is diagnostic, treatment selection, or better predictive analytics? From your research, where you think is more excitement and opportunity for success using machine? Yeah,
I want to understand your question. So you're
based on your experience and research with the models, where you feel more excitement, like is there better opportunity on the diagnostic side for disease, presence of disease, or better on the prediction side, analytics, predicting outcome or treatment selection? Where do you think is more likely we will have success, depending on the complexity of the model and the time it takes to develop those?
Yeah, that's the question that makes me to think the most among all questions I got from invited seminars. Yeah, that was an excellent question. So I think to actually solve real world problems in clinical medicine, it's not just about good accuracy. And then you just saw an example, whenever the machine learning model performs well in terms of the prediction accuracy, it may be that they are looking at wrong things, right? So, and then I think, reliability, trustworthiness of this machine learning model is something we need to improve. And then, first accomplishing good prediction accuracy, we think that we are there, but we may not be. And then also, training data can have a lot of confounders. It could be from some subpopulations too dominantly. And then, so I think, we first need to achieve good, predictive accuracy, or, the intended job, but in the right way. And then we need to have a way to detect when it doesn't. Because testing using, cross-validation test, or, even external data testing is not, they are not enough. Yeah, I'm not sure if I answered your question, but I think the number one importance is intended job. And also there should be a way to detect it when it doesn't, the system doesn't.
Thank you, Sven. Great.
Yeah, it's harder than we just saw the example, right? It's a COVID example.
Dr. Harefield has another question. So he asks a tangential question. Could your models assist physicians in understanding their own justifications, rationalizations, and explanations for their own daily therapeutic decision-making? So physicians are making life and death decisions daily without such models.
I know, yeah. I think this is important, incredibly important, and it's not really accomplished by the system a researcher like me builds. It needs to be, clinicians should also get used to this. You know what I'm saying? So interaction is really important.
Well, the thing that concerns me is these people work extremely hard. They have phenomenal memories. They have a lot of experiential mileage. They're making these decisions in the trenches, life and death decisions, but they don't have access to your system. How do they justify, how they rationalize and feel confident in making these life and death decisions? And can your system help them do their job with greater confidence?
Yeah, I think if clinicians have access to, what this, machine learning models prediction will be given certain set of data, and then why, I think it will certainly help. But what I'm saying is that it's not the end. It's not the only things that help making this happen. It needs to also be the training of the clinicians such that they get used to this information, and then, education of how these, these assists were generated by the AI system. Does that make sense? Yeah, so my short answer is that yes, it can, but it's not, we need more than that.
Thank you. Dr. Harefield says thank you. So thanks so much for, I don't see any more questions in chat. If any of the other participants have questions, please unmute and ask directly, or you can enter them in chat.
Thank you, Dushyant.
Okay, so it doesn't look like there's any more questions. So thanks, Dr. Lee, for an excellent presentation. And we yeah, I really enjoyed it, and I'm sure everyone really enjoyed it. And thank you so much for an excellent presentation.
Thank you for inviting me. I really enjoyed giving a seminar too.
Sure.